NICS, which stands for Non-Invasive Chromosomal Screening, is a non-invasive PGT-A technique that tests the cell-free embryonic DNA secreted by embryos during the embryo culturing process to non-invasively screen for chromosomal aneuploidy in embryos and select healthy embryos for implantation. To avoid maternal cumulus cells and other impurities during embryo culturing, which would interfere with the NICS test results, and to ensure the accuracy of the NICS test results, we prepared this video to give instructions of the standard operating procedures related to NICS IVF embryo culturing and culture media collection. We will provide detailed demonstrations and explanations to all the key operating procedures related to NICS culturing and embryo medium collection. To implement NICS tests, IVF centers should be capable of performing egg retrieval, ICSI, embryo culturing, and embryo vitrification. The required reagents for NICS include mineral oil, gamete medium, fertilization medium, cleavage medium, blastocyst medium, hyaluronidase, embryo vitrification buffer, and a Yukon sample collection kit. The required consumables for NICS include 4-well dish, operation dish, culture dish, holding an injection needle, a Pasteur pipette, and denudation pipette. The required equipment for NICS includes a microscope, a laser system, a micromanipulation system, and a tri-gas incubator. Prepare denudation pipettes using Pasteur pipettes, or purchase denudation pipettes directly from commercial vendors. Prepare 3 denudation pipettes, each with different inner diameters for later uses. After digesting the maternal cumulus cells with hyaluronidase, use denudation pipettes to remove maternal cumulus cells, starting from the denudation pipettes with the largest inner diameter and finishing with the denudation pipettes with the smallest inner diameter. While denuding the embryos, please repeatedly aspirate the embryos in and out of the denudation pipettes until maternal cumulus cells are completely removed. After denuding the embryos, observe the embryos under a microscope to evaluate if maternal cumulus cells have been completely removed or if there are still remaining maternal cumulus cells. Please try to remove maternal cumulus cells as completely as possible. Remaining maternal cumulus cells that were difficult to remove can be denuded later before medium change. After embryo denudation, please perform ICSI under a micromanipulation system. (18 to 20 hours after ICSI, observe embryos for fertilization. If fertilization is successful, observe embryos on day 2 and day 3 for embryo cleavage. Continue culturing the embryos that finish the embryo fertilization and cleavage. There are 2 methods for culture media collection. Each IVF center can choose either method for culture medium collection based on its resources, demands, and preferences. The first method is to collect culture medium from day 3 to day 5 after the medium change. This is suitable for sequential-based culturing, with medium changes taking place on day 3, for IVF centers accepting single microdroplet culturing from day 3 to day 5. The second method is to collect the culture medium on day 5. This is suitable for one-step-based culturing or IVF centers not accepting single microdroplet culturing before day 5 after the medium change. This method will involve the use of a laser system to perform assisted hatching. We will demonstrate each medium collection method. For method 1, you will collect the culture media from day 3 to day 5 after the medium change on day 3. You will prepare a culturing dish, washing dish, and blastocyst dish. You will prepare a blank droplet in the blastocyst dish. Each embryo has 3 corresponding washing droplets in the washing dish. You will then observe the day 3 embryos. If there are a few remaining maternal cumulus cells attached on an embryo zona pellucida, as shown in the picture below, denudation should be performed to remove the remaining maternal cumulus cells. If there are 5 qualified cleavage-staged embryos, 6 pipettes are needed, 5 for embryo transfers and 1 denudation pipette with the smallest inner diameter for denudation. First, use 1 denudation pipette with the smallest inner diameter to remove the maternal cumulus cells of the 5 day 3 embryos. After repeatedly aspirating the embryos in and out of the denudation pipette, observe carefully to make sure that the maternal cumulus cells are completely removed. Using pre-labeled and pre-numbered glass Pasteur pipettes to transfer each denuded day 3 embryo from the culturing dish into the corresponding washing droplet in the washing dish, try to minimize the carryover of medium from the culturing dish to the washing dish. After transferring each embryo to the washing dish, wash each embryo serially in the 3 corresponding microdroplets for each embryo by pipetting. After washing the embryos, transfer each embryo from the washing dish into the corresponding single microdroplet on the pre-labeled blastocyst dish. Use a unique transfer pipette for each embryo. After the medium change on day 3, incubate the embryos in the blastocyst dish in the tri-gas incubator to day 5. Transfer each qualified embryo to the vitrification dish for vitrification using embryo vitrification buffer. The remaining culture medium is collected. We are going to demonstrate the culture medium collection process. After transferring the embryos to the vitrification dish, collect the remaining culture medium for each embryo on the blastocyst dish using a unique pipette to pipette the medium up and down 3 times carefully and then transfer it into a pre-labeled 0.2 microliter PCR tube from Yukon sample collection kit. Store the collected culture medium samples at -20 degrees Celsius until the courier is ready to pick up the samples. The second method for culture media collection is for collecting culture media on day 5. You'll prepare a transfer pipette, a culturing dish, a washing dish, and a blastocyst dish. Prepare a blank droplet in the blastocyst dish. Each embryo has 3 corresponding washing droplets in the washing dish. For each embryo, prepare a 10 to 15 UL droplet in the blastocyst dish and equilibrate the blastocyst dish in a tri-gas incubator overnight before using the blastocyst dish. Observe the blastocyst on day 5 and document the embryo grading of the blastocyst. Choose blastocysts with expansion degrees of level 3 or above for subsequent operations. Transfer the blastocyst with an expansion degree of level 3 or above to the corresponding washing droplets on the washing dish for washing. Each embryo has and is washed in 3 separate washing droplets on the washing dish. After embryo wash, transfer each washed embryo into the corresponding single microdroplet on the blastocyst dish. Under a high magnification microscope, use a laser system to generate a hole on the zona to start blastocyst shrinkage. After complete shrinkage, the blastocyst will be placed in a tri-gas incubator to incubate for another 4 to 6 hours to allow sufficient release of embryonic DNA into the culture medium. Transfer each qualified embryo to the vitrification dish for vitrification using embryo vitrification buffer. The remaining culture medium is collected. We are going to demonstrate the culture medium collection process. After transferring the embryos to the vitrification dish, collect the remaining culture medium for each embryo on the blastocyst dish using a unique pipette to pipette the medium up and down 3 times carefully, and then transfer it into a pre-labeled 0.2 ml PCR tube from Yukon sample collection kit. Store the collected culture medium samples at -20 degrees Celsius until the courier is ready to pick up the samples.